Okay, I think we can get started. I want to thank everyone for attending this morning, our webinar on use of our devTOX quickPredict assay as an alternative method under the revised ICH S5R3 guidelines. If you have any questions throughout the webinar, please enter them into our Q&A box and we will answer those at the end. For those of you who don't know, Stemina is a small business located in Madison, Wisconsin. We established our operations in 2007, and we really designed our facilities to be a state-of-the-art mass spectrometry and stem cell culture laboratory. There's really two sides of our business. The first is our toxicology business, which I'll be talking about today, which looks to use human stem cell models for studying toxicity and disease. The second side of our business is our diagnostics business, which looks to identify biomarkers and different diagnostic methods for identifying different neurodevelopmental and neurological disorders. Particularly, we have a special focus on autism spectrum disorders. The two in vitro toxicology assays that Stemina currently offers are our devTOX quickPredict and our cardio quickPredict assay. Both of these are focused around the idea that we're going to make an exposure-based prediction of toxicity, so that a compound, isn't just positive or negative, but it can be positive at certain concentrations and negative at certain concentrations. They both use a targeted metabolomics platform, and the devTOX quickPredict assay uses human pluripotent stem cells, and our cardio quickPredict uses cardiomyocytes differentiated from them. Obviously, the focus of our talk today will be on our devTOX quickPredict. So when we started designing these assays, we really felt that human pluripotent stem cells offered a unique opportunity for screening compound safety using a human endpoint. And this is important because it really allows us to eliminate species-specific differences in toxic response. As you know, there are many limitations with in vitro assays, but by using these human cells, we can at least reduce one of those limitations. So the devTOX quickPredict assay uses our embryonic stem cells or induced pluripotent stem cells in their undifferentiated state. And we selected to do this undifferentiated state because it really allows us to pick up multiple types of developmental toxicants that are going to cause many different malformations. So we're not just focused on one lineage. As I had mentioned, the cardio quickPredict does use cardiomyocytes that have been differentiated from iPS cells. And the goal is that we're going to develop and provide assays that allow our sponsors to understand compound risk profiles faster, looking at multiple exposure levels, which allows us to make that exposure-based prediction, using a very small amount of compound and at a much lower cost than what the traditional in vivo studies would be. So just a brief overview of how this assay was developed. We initially developed it using human embryonic stem cells, which are derived from the inner cell mass of a blastocyst and have this potential to self-renew in culture and remain pluripotent for many passages. Once induced pluripotent stem cells became available and became a feasible option, we optimized the assay that we had developed with the ES cells for use with these somatic cell, dedifferentiated somatic cells. And the process is that there are many different ways to dedifferentiate these, but in the end, the iPS cells really have the same properties that an embryonic stem cell has, where they are pluripotent in culture and can self-renew. To identify the biomarkers that we use in our targeted assay, we exposed the cells to a training set of 23 well-characterized compounds. Many of these are drugs and vitamins that have well-defined human developmental toxicity outcomes, because we wanted to focus on what the effect was in humans. So we measure changes in the secretome. So we're looking at how the cells are interacting with their media. What are they secreting into the media? What are they consuming from the media? And how does that change when you treat them with a developmental toxicant, and making sure that that doesn't change when you treat them with a non-developmental toxicant? And so we did this many times and looked for the most reproducible biomarkers, and then looked to see, once we had a set of candidate metabolites, could we narrow that down to a limited set that was most predictive and defined and had important biological roles during embryonic development. And through this, we were able to find 2 metabolites that predicted developmental toxicity with as high of accuracy, sensitivity, and specificity as when we used a much larger model containing many unknown features and metabolites. And so these 2 were ornithine and cystine, and they're used as a ratio in our devTOX quickPredict assay. Both of these are essential for normal embryonic development. Ornithine is secreted by the cells into the media, and most often you will see a decrease with toxicity, whether this is because there is a cytotoxic effect and the cells are dying, or there are a number of compounds that will cause a suppression of ornithine secretion without cell death based on changes in the biochemical pathways producing ornithine. There are a handful of compounds, most notably the retinoids, that will cause ornithine to increase with toxicity, and this is directly linked to these retinoids' mechanism of action, where they will inhibit the ornithine decarboxylase enzyme. The biochemical pathways that ornithine is most involved in is the urea cycle, and it's also a rate-limiting metabolite for polyamine synthesis and pyrimidine synthesis. Unlike ornithine, cystine is actually a media component, and when the cells are happy, they are consuming about 75% of the cell or cystine from the media. However, when you treat them with known developmental toxins, what you'll see is that they stop consuming this cystine, or stop transporting it from the media into the cell. And you'll see this increase relative to the controls because there's more cystine in the media from the cells not taking up as much. The main function of cystine is in the cystine-cysteine redox cycle, which is a major player in regulating oxidative stress. And once taken into the cell, cystine is converted into cystine, which is involved in glutathione metabolism. And as I had mentioned, both of these are essential for normal embryonic development. And so they need to be tightly regulated for normal proliferation and differentiation. And so you can imagine if either of these processes are dysregulated, that can lead to embryonic death or malformations. The process that we use is we'll take undifferentiated either HES cells or IPS cells. The HES cell line we use is one of the original lines developed by James Thompson in 1998, the WA09 line, and it's really one of the most published HES cell lines out there. And the IPS cell line we use is a line that's commercially available from ATCC. And we'll plate them into a 96-well plate, where each plate will include positive and negative controls, as well as reference controls, and then 8 concentrations of each compound. And so the 8 concentrations will typically span from about 0.1 to 300 micromolar, depending on compound solubility and what else is known about the chemical we're testing. These positive and negative controls are included to ensure that the cells are metabolizing ornithine and cystine in a manner that is consistent with how the assay was developed. And this is important to make sure that we can maintain the same predictions reproducibly year after year. After we expose the cells for 48 or 72 hours, depending on the cell type, we will then collect the spent media and remove all of the proteins and analyze it with a targeted LC-MS method that includes labeled internal standards for normalization. That allows us to use the cells for a cell viability analysis and additional downstream processes, because the cell viability assay we use is non-destructive. And then we'll take the data from these two endpoints and fit them with nonlinear models to identify what concentration, at what concentrations they are changing. So if you take a closer look at that graph, as I mentioned, the idea is that toxicity is a function of exposure. And so what we're interested in is, when is this OC ratio changing, and at what point does that change signify developmental toxicity? And to identify that, we have developed what we term our developmental toxicity threshold, which was set using that original training set of 23 compounds and comparing the response in the OC ratio to the therapeutic Cmaxes or Cmaxes that were known to cause developmental toxicity in humans. And the goal was to set this threshold so that it balanced sensitivity and specificity. So what we're looking for is, when does this OC ratio cross this threshold? And that concentration where it crosses is really going to be your developmental toxicity potential concentration, or your DTP. And that concentration can be compared to cell viability to understand mechanisms, whether or not the OC ratio is changing independent of cell viability. Are the effects we're seeing occurring concurrent with cell viability? And this allows us to really set up toxic and non-toxic windows. So everything before that, where that ratio crosses the threshold, would be considered to be non-developmentally toxic, whereas everything, all the concentrations above where that ratio crosses, are considered to be your developmentally toxic window. And we've tested this with over 100 compounds with a very diverse set, looking at pharmaceuticals, agrochemicals, cosmetics, industrial chemicals, et cetera, and compared all of that data with published human and in vivo results. We have two different prediction models we use for evaluating the performance of the assay. The goal is to really compare it to exposure data whenever possible. So if there is in vivo exposure data, whether it's human or largely rodent, we will compare the DTP concentration to that therapeutic or low L Cmax that is known to either be developmentally toxic or non-toxic, and make a prediction based on whether or not that DTP occurs before or after those concentrations. However, we do recognize that often this information isn't available early in during chemical development, and it's also not available for a large number of agrochemical and industrial chemicals. And so we also use that training set to set a threshold for prediction based on how much, at what concentration can we separate developmental toxins from non-toxins, and using a threshold of 65 micromolar, we'll make a prediction as to whether or not something is toxic or not. And so using these models, looking at the 124 different chemicals, we had an accuracy of 87%, with a sensitivity of 88 and a specificity of 86. And this really gives us a nice balanced positive and negative predictive value. And one of the things that I think the devTOX assay, where it really excels, is that you can be pretty confident that when something is predicted as positive in our assay, it's going to be positive in vivo. So as I mentioned, the focus of today's talk really is on these new ICH guidelines that allow for alternative assays to be used during pharmaceutical development. And within Annex 2, they provide a comprehensive description of what needs to be included when you submit data from one of these assays for one of the proposed scenarios. And so the information that they are saying needs to be provided to the regulators is really a lot of background information on the alternative assay and predictive model, including a description and justification of the predictive model, which species endpoint you're looking to predict, whether it's rat or rabbit, and which endpoint, which for our assay is malformations in embryo fetal lethality. What is the biological plausibility of this model? The definition and justification for the threshold, and description of the model for determining positive and negative outcomes. So as you can kind of see, this is all stuff that I have just described. And so you can see that most of this all of this stuff has already been developed and is available for use with the devTOX quickPredict. Now, for assessing the performance of the model, they want you to use a range of biological and chemical domains that are justified for an assay's context of use. So they're going to look at assessing the sensitivity, specificity, positive and negative predictive values, as well as the reproducibility. And then you also will need to provide a list of the training and test sets, and if possible, including a number of human teratogens that were not correctly detected in vivo by rat and rabbit, such as thalidomide or Accutane. And then they provide 29 chemicals in the ICH guidelines that have in vivo exposure data. But if you're you're able to include a number of other compounds as well, you just have to be able to provide that same in vivo exposure data and the data sources for where you obtained that from. So what I'm really going to focus on here, the rest of my presentation, is this section, since we already kind of talked about this first section. And so as I mentioned, in the ICH guidelines, they provide a list of 29 positive reference drugs. And in addition to these positive controls, you need to be able to include some negatives to show selectivity of the assay and that you're not just predicting everything as positive. With all of these, they provide the in vivo information, and they also provided 3 negative controls. Now with devTOX, we have actually tested over 110 pharmaceuticals, including 31 of the 32 chemicals that were included in the ICH. The only one we have not tested is ibuprofen. The data for 65 of these pharmaceuticals is available right now, and that's part of what I will summarize in the remaining portion of the presentation. And the data for the other about 50 pharmaceuticals will be available this fall as part of a joint publication we're doing with the Tox21 cross-partner project we did with the FDA, EPA, NICEDM, and NCATS. And so for all of these about 115 compounds, I've done literature searches to really identify what are all the rodent and rabbit NOELs and LOELs, and what are all the corresponding Cmax values for understanding at what concentration these drugs were positive or negative in vivo, and really being able to use those for understanding any responses that we observe in the assay. In addition, I've gone through and looked up what are the different target receptors, enzymes, etc., for each of these drugs, and whether or not they are expressed in the human pluripotent stem cells. And that's important because that will really help us understand what is the applicability domain of our assay. So looking at just the performance of the devTOX quickPredict for those 65 pharmaceuticals, the balanced accuracy was 90% with a sensitivity of 87% and a specificity of 92%. And these predictions were all made by using that comparison of the DTP concentration from our assay with the in vivo Cmax concentrations from rodents, rabbits, or in some cases humans. We had, again, a very nice high positive predictive value of 94% and a negative predictive value of 83%. When we look at whether or not, how concordant our assay is with the in vivo, so how many times does the prediction agree, whether or not it's right or wrong compared to humans, we were 80% concordant for 65 of these chemicals with rodents, and there was only data for 53 of them in rabbits, and for rabbits, we were 85% concordant. And so you can see, one of the things we need to think about is whether or not are we looking to predict and use this model in place of a rodent in vivo test or in place of a rabbit in vivo test. And you can see our concordance is very high, regardless of which species you want to use. Another thing that we wanted to do was look at how accurate were we at predicting the human response. So for 48 of the drugs, they are either known to be developmentally toxic or non-toxic in humans, or they have mechanisms that have been, if there's not data for that specific drug, it has a mechanism that is known to be developmentally toxic. And so when you look at our concordance and accuracy at predicting the human response, you can see we actually outperformed the rodent and rabbit tests with a balanced accuracy of 94%. Our sensitivity was just as good as rodents at 93%, and we had a little bit higher specificity at 95% compared to 80 and 87. One of the things as well that you want to make sure is that, if you're using these in vivo exposures for prediction, you want to make sure that your assay is going to correlate with the in vivo exposures for developmental toxicity. And so for a subset of these chemicals that had positive and negative exposure data that I took from the Daston et al. Publication back in 2014, I compared our DTP concentration to the positive exposure value. And you can see that the assay was highly correlated with the in vivo positive effect exposures. The exception would be, all-trans retinoic acid, which is much more potent in our assay than it was in rodents, which is consistent with what you would expect based on the fact that humans are known to be more sensitive to the retinoids than rodents are. And then the other outlier would be dihydroartemisinin, which is the active metabolite of artesunate, which was not as potent in our assay as it was in rodents. In addition, we've looked at thalidomide and two of its analogs, which, as many probably know, was not predicted. Thalidomide itself was not predicted well by the rodent model back in the 1950s. And you can see it is being predicted and picked up in the devTOX quickPredict assay. And both of these two analogs that are suspected to be human developmental toxins and had discordant data between rodent and rabbit are both picked up as well and have similar potencies and to thalidomide, and thalidomide is actually a bit more potent than thalidomide is. So when we're thinking about the applicability domain, so you can see as an overall evaluation, devTOX quickPredict does an excellent job at predicting pharmaceuticals. Now, that doesn't mean that it's going to predict all, I'm going to say that it predicts all pharmaceuticals with great accuracy, because there are going to be a number of things that may not be picked up in the assay. And so if one way you can try and parse out, where is the assay most suited for application is by looking at the different pharmacological classifications, and then even getting a little bit granular, looking at the specific targets themselves. Overall, the assay picks up the different pharmacological classifications with a high sensitivity. You can see, it picks up DNA modifiers, enzyme or kinase modulators, nucleoside modulators, second messengers, and transcription modulators, all with, 100% sensitivity. There is a couple of cases in the enzymes where we are not identifying that compound as being developmentally toxic. And you can see the receptor modulators, we have about 50% sensitivity for the 4, predicting 2 out of the 4 positives. And one example of those receptor modulators where we did not correctly identify the developmental toxicity was bosentan, which is an endothelin receptor antagonist. And this is consistent with the data that was published by the EPA last year, where the endothelin receptor antagonist was an area that they identified the assay as being part of, the negative domain for the quickPredict assay. And you can see bosentan does cause an effect in the assay, but not until you get to concentrations that are significantly above where that rodent Cmax is in vivo. One thing to note about this is that this compound was not concordant between rodents and rabbits. So in rabbits, they did not have an effect. There was no effect observed. And so this would be where our assay correlates well with the rabbit assay over the rodent. And the effects in humans are really not occurring typically until that late 3rd trimester. And so this would be, again, outside the applicability domain of the assay. A couple of examples we're showing with enzyme modulators, where you can see the assay is able to separate the positive and negative compounds is with acyltivomir and lovastatin. Please ignore that on the graph because that was an incorrect label. But you can see there's Tamiflu, which is also oseltamivir, is not showing any effect at the relevant concentrations, whereas lovastatin is showing a nice effect before you reach that rodent Cmax for the lowest observed effect level for developmental toxicity. And again, both of these are affecting enzymes that are expressed in the cells, whereas with bosentan, the expression was minimal, if at all. So in addition to looking at the reference compounds, there are specific scenarios where the ICH and the regulatory bodies feel that alternative assays could be applied today. And so one of those is for pharmaceuticals that are expected to be embryo-fetal toxicants based on their mechanism of action. And this is probably the lowest hanging fruit, I think, the easiest one where you can use an assay to, predict whether or not something's going to be positive. And so Stemina has submitted a letter of intent for the FDA's Biomarker Qualification program, starting with this scenario, with the goal of once we do our full qualification with this scenario, we can move on to some of the other ones that I'll mention. And the specific context of use that we've developed for the assay is that the ratio of ornithine to cystine can be used as a safety biomarker for detecting human developmental toxicity potential in vitro using human pluripotent stem cells at the non-clinical stage for small molecule drug candidates expected to be embryo-fetal toxicants as described in Annex 2. And the goal, as you can see right now, we're starting with something that is going to be early human development and small molecule drug candidates. So I think the assay does have application for biologicals, depending on what the target of that biological is, but we don't have enough experience right now to move down that route in the qualification package. So we're going to start with the focus on small molecules and then expand as more data becomes available. The other scenarios are for pharmaceuticals that are intended to treat severely debilitating or life-threatening diseases, or pharmaceuticals that are intended to treat late life onset diseases, as well as possibly deferring in vivo testing to include women of childbearing age in the clinical tests as part of an integrated testing strategy. And so with each of these three scenarios here, they provide different EFD testing strategies for utilizing these alternative assays. And, the one thing I would take away is that, in some cases, you are actually able to eliminate any use of in vivo studies when your assay is predicting a positive. So the decision tree for using and applying devTOX quickPredict for compounds that are expected to be embryo-fetal toxins is that, the first question you would ask, is your compound expected to be an embryo-fetal toxin? Yes. Is it within the applicability domain? So do we have data showing that the mechanism is that is expected to be fetal, an MEFL, is picked up with the assay? If it's not, then we would recommend either using a different in vitro assay or conducting your traditional in vivo studies. But if it is, you could then evaluate your compound in devTOX quickPredict, and if it's positive in the assay, there would be no additional assessments required at this point by the regulatory agencies. If the results were negative or equivocal, then you would want to perform your in vivo studies to confirm that something is negative for MEFL. So one example, I'm going to provide just a couple of examples of what data could look like and what you could do with that data is, in this case, you test your compound and we don't see any effect in the assay. And if that was the case, you would go down this route of having to perform your in vivo studies. Whereas if you tested your compound and we saw a nice response in the OC ratio, you could then use this other branch where you're done with your in vivo testing at that point, or you're done with your testing at that point and don't have to move into the in vivo realm. So what can you do with this in vitro concentration, and how does it relate to interpreting it in terms of the in vivo data? The most simple interpretation, I would say, is comparing this concentration with your expected therapeutic exposures, if you know what those are going to be, to help interpret, is this effect occurring at concentrations where it's going to be relevant in humans, or is it occurring at concentrations that are, say, are 100-fold higher than where you would expect the human effect to be? The other interpretation is you could use in vitro to in vivo extrapolation to really do reverse dosimetry to predict the equivalent administered dose from this TTP concentration. So if you have a DTP of 0.5 micromolar, you can use the different tools out there for PBTK modeling and predict what is that dose equivalent to in humans in a mg per kg per day, and is that dose then within the realm of what humans would be expected to take. Or you can predict your rat in vivo doses using these models and interpret the data in terms of what other toxicity data you may have, whether it's for, acute toxicity, things like that, to understand, are these effects going to occur at concentrations where no other toxicity is expected to be observed. So with that, to kind of summarize what I've talked about, devTOX is an accurate and reproducible assay for predicting human developmental toxicity. It's able to identify a wide range of mechanisms, but it may not be applicable in all cases. And in particular, the first applicability area that I expect to be is outside of our applicability domain, are for targets that are not expressed in pluripotent stem cells. The second conclusion is that the DTP concentrations that we predict are strongly correlated with those in vivo positive exposures. So your Cmaxes from your lowest observed effect levels for developmental toxicity. You can combine the data from DTP, from the devTOX assay, with IVE to estimate your equivalent administered doses and better understand how these DTP concentrations relate to the expected therapeutic dose or in vivo doses in rodents where other toxicities may be observed. And lastly, I wanted to stress that all of the information and data for using devTOX quickPredict as an alternative assay is available, and it can be submitted as part of an NDA for pharmaceuticals that fall into those three specified scenarios. As I mentioned, we are working on doing going through the biomarker qualification program to pre-qualify the assay for use with compounds that are expected to be embryo-fetal toxicants. And that's really more, the idea of that is that it would then provide, once we finish that process, all of the necessary qualification data would be there within the FDA, and sponsors wouldn't have to submit it individually, hopefully. But by doing it, but that doesn't mean that you can't use it now, I guess is what I'm trying to say. It's just a little bit more work, and we're happy to do that with you and help put together the necessary information for you to use the assay under the ICH guidelines. So with that, I just want to thank everyone for attending this morning, and I will take any questions that you may have.
Great, Jess. Thanks for that. We do have a couple of questions in the chat, and please feel free to ask additional questions or raise your hand and you can ask the question live. The first question is, how do you calculate the balanced accuracy?
So the balanced accuracy is calculated as a sensitivity plus specificity divided by 2. There's multiple ways that you can calculate it, but that's the one that I've seen used most often. So it's really, the idea is that especially when you have an uneven number of positives and negatives, you can balance out. You're not just overloading that accuracy with one or the other.
Okay, great. Another question in the chat is there any evidence that your assay would detect receptor-mediated neurodevelopmental dysmorphogenesis, such as an impact on synaptogenesis?
So we do pick up the, developmental neurotoxins very well. There's a number of compounds that we've tested that cause that effect in vivo or in other in vitro assays. And so, we're not going to be able to tell you whether or not it's going to impact synaptogenesis, but we will be able to tell you that it's positive. And so for most of this type of a receptor-mediated effect, we do see that with the assay.
All right, great. Another question, have you looked at the specificity of the biomarkers to developmental toxicity? For example, do these biomarkers change in other cell types besides human embryonic or induced pluripotent stem cells?
Yeah, so we've looked at, we've done metabolomics for numerous cell types over the years. And the one thing we found is that, each cell type, each toxicity is really going to have its own set of biomarkers. And so, when we look at, say, cardiomyocytes and looking at cardiotoxicity, none of the biomarkers that were important for developmental toxicity are not being picked up as important predictors for cardiotoxicity, whereas the biomarkers, and the opposite is true, where the biomarkers for cardiotoxicity aren't showing up as important markers of developmental toxicity. And the other evidence of that is that there are a number of pharmaceuticals that we've tested that are known cardiotoxicants, but they're not developmental toxicants, and they're not picking up, they're not causing changes in the pluripotent stem cells that are indicative of developmental toxicity. So you can, it really shows that these markers are highly specific to this endpoint that we're looking to measure.
Okay, and so maybe you partially answered this other question, but have you tested other chemicals with no developmental toxicity, but other target organ toxicity?
Yeah. So that's part of it. We have tested a number of, like I said, compounds that are known to be cardiotoxic, and I can't tell you, there's none of those that I've seen that cause a developmental toxic effect unless there is known developmental toxicity associated with them, like doxorubicin is a developmental toxicant as well. But something like tegaserod, which is a potent cardiotoxicant, it's negative for developmental toxicity, and it's negative in our assay. We've also tested some different neurotoxicants that are not necessarily developmental neurotoxicants, but they cause neurotoxicity later in life, and most of those are also negative in the assay.
Okay, great. How do you think about this assay in the sort of reduce, refine, replace 3Rs strategy that we talk about? Can you give a couple of examples where you think that the 3Rs can be accomplished using this assay?
Yeah, so I think obviously the easiest way would be reducing animal use, using this assay early on as a screen for identifying which compounds are most likely to be developmentally toxic, and only moving forward with your most promising compounds that aren't going to cause developmental toxicity. You can really reduce the number of animals that are being used because you're not having to screen as many or test as many for developmental toxicity. And one of the great things about this new ICH guideline is it really, before it was hard to say when an in vitro assay would be able to, say, replace an in vivo assay. But with the new guidelines, you can really use a positive effect in your assay, in the in vitro model, would replace needing to do any in vivo testing to identify a positive effect. So those are really the two main ways that I can see using it to reduce and replace animal models.
All right, and I know that we've developed a number of interesting sort of specific case uses for using the assay. Can you talk a little bit about some of the, for example, smoke condensate systems that we built, and also the ability to test gaseous compounds?
Yeah, so in efforts to really expand the utility of this assay and outside of your standard single compound types exposure systems, we have worked with a couple of different tobacco companies at testing different types of mixtures that are produced from either smoke condensates or are produced as an aqueous solution when the smoke is bubbled up through media or PBS, and tested for developmental toxicity using adding those types to the media. And then testing for whether or not that mixture is going to be developmentally toxic. And we had a publication last fall that describes, the initial application of the devTOX quickPredict assay, where we looked at the, traditional cigarettes and compared that to some of the newer generation products that are, either tobacco free or tobacco containing, and found that when you test the, say, liquids, or when you test the smoke from the e-cigarettes, it's not as toxic, and you don't see an effect in the assay at the same level where you see an effect with the traditional cigarettes, showing that, demonstrating that the e-cigarettes are really likely to be less toxic. Another application that we developed was for testing compounds that are highly volatile and may not maintain that solid state at room temperature, or even at the temperatures in the incubator used for culture. And that is where we looked at an exposure system that uses Tedlar bags, and then we can expose the cells. We'll seal the plate in one of those Tedlar bags and add a specific amount of incubator air, so 5% CO2, back to the bag, and then add a specified concentration of that gaseous chemical to the bag, and expose the cells in that state to the gaseous or volatile compound. And we've shown that using that system, we're actually able to pick up the compounds that are expected to be developmentally toxic, such as acetaldehyde, and we can also do studies to identify, how much of that gas is getting into the media, and what is the concentration being exposed to the cells versus the concentration that's in the bag itself.
Great, thanks, Jess. Just one other question. How do you use this in combination, for example, with other in vitro and non-mammalian systems?
So you could use this assay. There's a number of ways that you could combine it with other in vitro systems, and you could identify, which ones are going to be the most complementary. Where you test everything in both assays and use a consensus prediction. So say you combine this with the zebrafish assay, which ones are going to be a consensus, which ones are positive in one or the other assay would be considered a positive overall for prediction. And that, really will increase your sensitivity, but it could decrease your specificity doing it in that manner. The other way is using like a tiered testing approach, where you test with, say, devTOX quickPredict first, and then if it's negative in devTOX quickPredict, move on to testing in, say, the zebrafish or the mouse embryonic stem cell test, and seeing is it positive or negative there, and then either, moving down to another step, or, taking that as your answer. But the goal is really to find, which ones are going to pick up, what assays are going to pick up the areas and the mechanisms that are known to be developmentally toxic, that the devTOX quickPredict assay is, missing. And there is some data that should be, the EPA has presented on over the last couple of years, and hopefully that publication will be out in the fall or early next year, where they really looked at all of the different ToxCast models and all of the different mechanisms to identify, which assays are going to be the most highly predictive, and which combinations are going to be the most complementary. That would really be the ideal situation, where you're, substituting or, adding in additional assays that are, specific to mechanisms that, say, devTOX is missing.
Okay, great. Are there any other questions from the attendees? All right, I do not see any additional questions in the chat, and I do not see a hand up. So thank you very much for attending. And Jess, thank you for a very informative presentation.
Right. Thank you, everyone, for attending, and thank you, Beth, as well.